My name is Wenying Jian. I work for the DMPK department of Janssen R&D, which is pharmaceutical company of Janssen Johnson. I'm leading a group to provide LCMS bioanalytical support for preclinical and clinical studies. My research interest is in application of advanced LCMS methodologies to large and small molecules, including some of the novel platforms such as liposomal product, protein drug conjugate, and peptide protein conjugate or fusion. LBA and LCMS are the two primary technology used for bioanalysis. LBA mainly for large molecule and LCMS primarily for small molecule and has been increasingly used for large molecule as well. Hybrid LBA and LCMS is to combine the advantages from both sides to overcome some of the limitations of analyzing large proteins in biological samples. I'm from LCMS background, so we know that LCMS provide unique advantage of being able to very specific detect structure information. On the other hand, it's very challenging to analyze large protein by mass spec, particularly in the background of thousands of proteins in biological samples. In this way, we can add upfront LBA procedure to clean up the sample, such as affinity capture, and then followed by LCMS detection. So we are using LBA to clean the sample up and LCMS for detection. So that's the basic principle of hybrid LBA LCMS. In our lab, the primary instrument for detection is mass spec. However, it's not easy to detect protein by mass spec. We use two primary platforms. One is bottom-up, the other is top-down. In bottom-up assay, we digest the protein into smaller peptide, which in turn can be relatively easily detected by mass spec. In top-down, we detect the protein directly by high-resolution mass spec without digestion. In both cases, we have upfront LBA procedure, affinity capture to purify the protein for analysis. LBA LCMS combines the advantage from both sides, being highly specific in detection thanks to mass spec, and being highly efficient in sample cleaning up thanks to LBA. In this case, we can achieve better specificity, selectivity, and sensitivity. Of course, it is subject to limitations from both sides. Same as LBA, hybrid LBA LCMS requires capture reagent, which may not be readily available and may take weeks or months to generate. And in terms of MS detection, mass spec is intrinsically less sensitive than LBA, which is one of the major limitations we have encountered in our work. And also, by combining the procedure from both sides, the workflow could be labor-intensive and time-consuming with added potential artifact and variability associated with multiple steps. I think hybrid LBA LCMS will be increasingly used for bioanalysis of large molecules as an important complement platform in terms of each of the technique alone. Very importantly, bioanalysis community need to accumulate knowledge and experience to understand the protein chemistry and the protein biotransformation to get an idea of their implication and impact on bioanalysis. I'm also hoping to see advancement in workflow efficiency, such as automation for faster and easier sample extraction and rapid digestion system when it comes to bottom-up approach. In terms of instrument, I'm hoping to see improved MS detection sensitivity and development in software for data processing, particularly for top-down protein analysis. Overall, I think hybrid LBA LCMS should be applied to broader scope of discovery and development and be more easily accepted by regulatory agencies.